So I'm very honored to have here with us Derek Aguiar. He's a professor in computer science department at the University of Connecticut, but I know him from really long time ago, and he inspired me and inspired my research during my time there in Princeton. And so I really welcome him, and the floor is all yours.
Thank you, Roberta. You give me too much credit.
It's the one that you deserve.
Oh, thank you. So thank you for the invitation. This is an honor and a pleasure to share some of our work that our group's done in the past year or so. Honestly, I didn't know that this problem existed 2 years ago. So to some degree, this is serendipity. But so it's a big team that worked on this project, this first project. And the way that this talk is going to go is we have this overarching project. And we're going to take a short detour to some more methodological components and then go back to this comp bio type project. But this was really led by the two co-first authors, Marzan Hosseini, my amazing PhD student, who's actually in the audience. And she's probably graduating this academic year, maybe, hopefully. And Aaron Palmer, who is not my student, but does a lot of work with my lab. And he is a postdoc just looking for a position. So anybody is hiring, he's an amazing computer science, statistics, machine learning researcher. So if I ask you to imagine the three-dimensional structure of DNA in your head, everybody probably has the same picture. The canonical double helix from Watson and Crick and colleagues. But actually, DNA can fold into different conformations. And this is the part that I wasn't aware of, 2.5 years ago. So the canonical double helix structure is called bDNA. It's the cartoon depiction on the top left. And these other structures, these are called non-bDNA structures. And there are several of them. Here, the structure is indicated in blue. And the motif, the DNA motif that is associated with them is given in black lettering. So you can have inverted repeats in the genome, and what these inverted repeats can do, and they don't always do this, but each strand can fold up and fold onto each other and bind to itself. So you have these cruciform-like shapes. Another really well studied structure is called a G quadruplex. And G quadruplex is also the name of the DNA base motif. It's essentially a bunch of guanines that have some spacers in between them. Okay, and they form these vertical sheets. The other strand is typically single-stranded DNA. And there are many of these other structures. So DNA bending, HDNA, slip strands, and they're all associated with some non-bDNA motif. Here I'm kind of showing you what these motifs look like. So especially the repetitive structures are all over the genome. G quadruplexes are less abundant, but they do exist everywhere. So the first thing, if you see some kind of unique pattern in the genome, the first thing that you would typically do is look to see if this is conserved across species, because that's at some hint that there's some functional relevance. So it turns out that non-bDNA motifs are conserved across species. So here on the right, there's just a plot where you have some statistics about G quadruplex positions with respect to likely locations for splice sites. So if you look upstream or downstream of an exon, so a likely location for a splice site, you can see that for 7 different species, G quadruplex motifs, DNA motifs are enriched at those sites. So that makes you think that this might be related to splicing. They're also prevalent in transcription factor binding sites, especially in human chromatin. Some evidence that they stabilize telomeres. And there's a lot of good evidence that they play a role in establishing centromeres. And there's even some work that we're doing or that we're starting to look into with neocentromeres. So centromeres that can form spontaneously. Okay, so there's a connection. And when you look at actually centromeric or telomeric DNA, you actually see a lot of these repetitive regions. So it would make sense that there might be non-B structures there. So if non-B is associated with basic biological processes, then it might also be associated with phenotypic processes. So here I'm showing an example that's specific to G quadruplex. So stabilization and induction of G quadruplexes seem to play a role in cancer-related pathology. So they associate with telomere maintenance problems, reduced oncogene expression. And also, there was this work that showed that even just independent of cancer, if you look at motifs, G-quadruplex motifs, and then you correlate that with level of mutability, so just mutations, the mutation rate is significantly increased in G-quadruplex motif regions. Okay, so seems to be related with mutability or maybe genomic instability. Okay, so now we've established some importance for these non-B DNA motifs. And by the way, this is mostly association work. It's really hard to find these structures, but maybe you'd like to find these structures in a genome. So what do most studies do? So most studies actually look for just co-occupation of a non-B motif with something that you're interested in. So maybe splicing or mutation rates, that sort of thing. But you can actually try to predict where the structures are. And there's two main ways to do that. The first way are experimental assays. So on the right, there was a study that used permanganate-seq S1 nuclease footprinting, which essentially is finding where is there single-stranded DNA. And if you have single-stranded DNA and it's a location that has a non-B motif, you can sort of intuit that there might be a structure here. So just going back for a second, like with G-quadruplex. So G-quadruplex is the strand that doesn't have the motif should be single-stranded DNA. So if you see single-stranded DNA in a motif site, it's good evidence for there being structure there. So it's not direct evidence, but no, tangential evidence. And they found that at least in the mouse genome, SMNB, that's just non-B motif regions in the mouse genome, there was single-stranded DNA in these motif regions anywhere between like 3 to 9% of the time. Okay. So this is important just to really stress the fact that non-B motifs, they're important for the purposes of this talk, let's just assume all non-B structures have motifs. It's not necessarily true, but just for this talk. Even though you have a motif, that doesn't necessarily indicate that you have structure. The other types of computational methods are actually very naive, which was sort of an indicator, which this is a good area to go into because the computational methods were pretty naive. Most of them, what they do is they'll do something with deep learning where they'll input some motifs and they'll make a prediction of whether or not there's structure, which is hard to do because there's no gold standard. There's no like ground truth. And even if there were ground truth, non-Bs are ephemeral and they can form and they can deform. But what most of them do is just look for motifs for these non-B structures and say, there's a putative structure here. Okay, there could be a structure here, but that's not evidence whatsoever because we see from the experimental techniques that non-B structures occupy a motif region 3 to 9% of the time, at least in the mouse genome. So the important takeaway from this is, again, they might not even be necessary, but just for the purposes of this talk, non-B DNA motifs are necessary, but they're insufficient. Okay? Just because we have a non-B motif does not mean we have the structure. Also, if anybody has a question at any point, just wave at me. Questions during the talk is fine. Okay, so there was this really nice genome research paper in 2018 where they showed that if you take PacBio sequencing and you calculate the time in between pulses, this is often done to find methylation marks, but if you calculate the timing between the pulses, this is called the interpulse duration. And in case you're not familiar with PacBio sequencing, you incorporate you have a template strand and then you incorporate bases, and the incorporation of the bases pulses, and you're able to call the base that's being incorporated into the strand opposite of the template strand. And so you typically just look for these pulses to call bases, but you can actually you can calculate the time in between these pulses. It's called the interpulse duration. If you look at these interpulse durations and you associate that with non-B motifs, just like this is what everybody does, right? You have something you associate with non-B motif sites, they find that there's a very distinctive pattern. Okay, so on the left, this is just a table reminding you of the non-B structures and their motifs. And you also have counts there. So you have a ton of microsatellites and much fewer zDNA motifs. And some of them don't have strong signatures, but G-quadruplexes do. Okay, so here is what they do is they compute a 100 base window around the DNA base motif. This is G-quadruplex. G-quadruplexes are about 20 bases long or so. So it's like 20 bases in the middle of this window. And then they're plotting the quantiles of the IPD signal, both for the forward strand that contains, or the strand that contains the G quadruplex motif, and the strand that doesn't contain the G quadruplex motif. Okay, so what do you see? You see that it seems to take more time to incorporate a base when, at least at the upper quantiles. So if you imagine that G quadruplexes are not forming that often, maybe they're forming just some small percentage of the time, then if we look at the upper quantile, we should see more distinguishing IPD values, if it indeed affects sequencing, between the non-B, which is in red, and the B DNA, or the helical DNA, in blue. Okay, so we see this really jump in IPDs, which means it's taking more time to incorporate the bases. On the reverse strand, kind of an interesting thing is it takes less time, which kind of makes sense because that strand's already unwound. If that strand is already single-stranded DNA, then the machinery has to do less work. Okay, and so this is called interval-wise testing. This is a non-parametric statistical test that basically compares aligned time series windows. And I believe this is a permutation test that compares whether or not in one set of time series, if you have over higher values or lower values with respect to some reference. So the reference here is the BDNA IPDs. And what we want to test whether or not it's higher or lower are the G quadruplex or A phase repeat or direct repeat. So the way that you can read this, and this is kind of, I don't love these plots because this is minus log 10 p-value for higher, and then this also says minus 10 log p-value, but it's just it's the different tails of the statistical test. You're just testing, the upper tail or the lower tail. And they find exactly what we kind of see with the quantiles. We see higher IPDs in G quadruplex. We see higher IPDs with respect to the statistical test compared to the BDNA, and lower on the reverse strand. And so there's some signature for these other non-B motifs as well. It seems like some specific signature. So we saw this paper. We were doing some work with others that were working with nanopore sequencing, and we really had two research questions. The first was, is there something similar to IPDs in Oxford Nanopore sequencing? And I didn't want to just do another sort of association study where we looked at the association of whatever this feature is. We wanted to actually build some computational models to see if we can predict structure. If this signature was actually direct evidence of structure, then let's try to predict the structure. It's just a tiny bit of background on nanopore sequencing. So nanopore sequencing, you have this nanoscale pore. You have this helicase enzyme at the top that's unwinding the DNA. Okay, it's pushing one of the strands through the nanopore. There's another enzyme that ratchets the strand through the pore. Sort of you can control the rate and the speed in which it goes through the pore. And then you have a current that's running through the pore. And depending on what bases are in the nanopore at any one particular time, and our work, we believe there's about, it's really hard to find this information, but we believe there's about 6 bases in the pore. It depends on the size of the bases, but about 6 bases in the pore. Depending on which 6 bases are in the pore, you get a disruption in the current, which you then use to base call. Okay, because different configurations of 6-mers will give you different disruptions in the current. So this is mostly used for base calling, DNA sequencing. So what is our hypothesis? What is the biochemical justification for what we're doing? We thought that if a non-B structure is forming upstream of the pore, then what's going to happen? It's going to hit the helicase, and the helicase can unwind non-B structures. This is this work from Boyer in 2013 that provides evidence that helicases can unwind non-B DNA, but maybe it unwinds it slower or maybe faster. Right? Maybe it's better at unwinding BDNA than it is G quadruplexes, for instance. So this is the direct evidence of structure that I think we can actually make a feature out of this. And so what's the feature? The feature is, how long does it take to ratchet the DNA through the pore? Okay. So this is just a cartoon of the process of base calling, and it's simplified because you're calling a 6-mer and we just have one base here. But Imagine you're annotating the base that's leaving the pore, for instance. And so you translate these current measurements, which are measured at a specific interval. So there's a frequency for measuring these. And when the current changes significantly, then you can call a new set of bases. Okay, this is how base calling in Nanopore is done. So what we do is we translate that into what we call translocation time. So how long does it take to translocate one base out of the pore and move one base in? And that's literally just the number of measurements that are taken per base move. And since we have the frequency, we can compute the time. This is what it actually looks like. I think this is one of the more well-behaved windows. But on the top here, you just x-axis are your measurements. Again, this is a regular interval. Y-axis is normalized current, and we translate these base called this base calling based on the current into times. Okay, so it seems like we have a feature. We don't know whether or not it's associated with non-B or if it directly indicates structure, but we want to try to use it to build computational models. Yeah, please.
A sense of like empirical statistics of the time. What's the minimum? What's the maximum? How much variance is there?
Yeah, that's a good question. So the minimum would be a single pulse, so it depends on the frequency of the sequencer. Whatever you set the frequency to, that would, I suppose, be the minimum. The maximum really depends on a lot of things. So for us, we think that we did a little bit of preliminary work to see if it's related to methylation, the times, and it doesn't seem to be, although the jury is still out on that. So I think the major indicator of how many current pulses you actually get is, I think, structural properties of the non-B DNA.
I understand. I'm just asking, like, what are the values? Is it a factor of 10, a factor of 100?
Yeah, I'm not sure. I mean, this is actual real data, so it seems like it can be anywhere between, a few to 20 or 30 frequency. And I forget actually, this should tell us what the frequency was. Yeah. So I'm not sure of exactly the statistics. That's a good question, though. And it's also a reason why Nanopore kind of doesn't do well when you have monomers. Because when you have monomers, then the 6-mer doesn't change. And so you don't really know when the 6-mer leaves the pore, because the next 6-mer that's coming in is exactly the same. So practically what we do is we do base calling, and then we map the reads back to the reference. And then we do this thing called resquiggling, which corrects this process. So if you were computing statistics for that, you would have to do that on the resquiggled stuff. Okay, so if you want to try to build computational models for this problem, then what are the challenges? So first of all, there's no machinery to calculate these translocation times. And so my students Marjan especially, had to go into these terabyte big files of nanopore sequencing and then find where the currents are and then use that to calculate the translocation time. It was a mess. So there's not a lot of support to do this just from a pipelining standpoint. The second challenge is that translocation time, at least if we're taking the same approach as the prior work and looking at windows, it's pretty high dimensional. We have 2 strands and maybe 100 base pairs per strand, so 200 dimensional vector. So the way that we handle this is we're going to use a deep neural network to generate embeddings. It's going to be a principal deep neural network, so I'll talk about that. And I know there's a lot of biostats people here, so you might actually like that application of deep learning. The third is we don't really have a label. What we have is a non-B motif label. We do not have a non-B structure label. Okay, and most of these motifs actually don't form structures. So we can't treat this like a classification problem. Instead, we treat this as a positive unlabeled problem. Another way you can think about this is novelty detection. So we're going to model the bDNA, the stuff that we know is double helical DNA. And then we're going to try to find things that deviate from double helical DNA. Okay? Because we can only really be sure of the bDNA class. And the last is we don't have ground truth. We have nothing to compare to. And we drew inspiration from the work in EQTLs. So with EQTLs, you're trying to associate a genetic variant with changes in expression. And there you don't have ground truth. You have no idea whether or not the genetic variant actually affects expression. So what do? You typically compute a hypothesis test for every single variant, for every single gene. Sometimes you restrict the region to variants just around a gene. And then you do some multiple comparisons corrections afterwards. So we're going to do something very similar. But the problem is we have these high dimensional translocation times, and we're going to embed them. But how do we do hypothesis testing in the latent space? Okay. So that brings us to a brief aside on some work that was published in iClear last year. And this work was led by, again, the same Aaron Palmer as was on the first slide, Chur, who was student of Stu Gima a long time ago. And then also Jimbo B, who was Aaron's advisor. And I show this image because we had our first annual graduate student Halloween event at UConn. And I drew inspiration from the Halloween event at Brown. And I was excited. You could see I was excited because I bought a costume for it, which was not cheap. And I think there was like 3 faculty there. So I'm working on it. I'm working on bringing Brown's spirit to UConn. Okay, so just in case you haven't seen these before, this is just a vanilla autoencoder setup. We have some input images, and the distribution of those input images. In this case, it's typically done with images. For us, it's going to be that 200-dimensional vector of translocation times. It comes from some p of x. Okay, it's unknown, but it's the true distribution of translocation times. We're going to push that through a bottleneck. So push that through an encoder, which is going to encode x down to z, and then a decoder that's going to blow z back up to the original data space, back to x. Okay. PZ is going to be our prior distribution on this latent space. Okay. And the goal here is, to match in this latent space, match the prior, and also ensure that reconstruction is the distance between X and its reconstruction is low. Okay, so let's go through just to show you the assumptions so we can contrast it with what we were doing. A couple of popular autoencoder variants. So a variational autoencoder is one of the first autoencoder architectures. And a variational autoencoder is going to map each individual point to the parameters of a normal distribution. Okay, so every single point is going to map to its own normal. This had some problems when they were doing this with images. They noticed that there were some blurring problems because you have these different normals that are mapped to some space and they're kind of overlapping with each other. But there, the PZ is multivariate normal, 0i, typically some isotropic Gaussian in the latent space. But what's important here is each point is mapping to its own normal. The Wasserstein autoencoder is based on optimal transport. And this maps what they call an aggregated posterior. So you're mapping a set of points to some prior distribution. Of course, you also have the reconstruction term. But here it differs from the variational autoencoder because you're mapping a mini batch of points. And you want the mini batch to appear to be similar to your P of C. Again, you have to fully specify the prior here, which is typically multivariate normal, 0i. Okay, so our model, it's called the goodness of fit autoencoder, or GOFE. It's similar. It's also based on optimal transport. But we extend a single specified prior, so multivariate normal 0i, to a class of priors. And our class is going to be the class of multivariate normals. In principle, it could be something else, but since everybody uses multivariate normals and there is good support for goodness of fit tests for multivariate normality, we chose multivariate normal. Okay, and you can sort of see that there's different multivariate normals there, and we can embed to any of those in the class. Okay, again, maybe I don't need to go through this because there's a lot of biostats people here, but we're going to be using goodness of fit tests. So goodness of fit tests is a type of hypothesis test whether or not some empirical distribution comes from a class of distributions. And so we're going to have 2 hypotheses. Our null hypothesis is that PY, just think about the embedding. The embedding comes from the class of Gaussians. G is going to be the class of Gaussians. H1, our alternative hypothesis, is that our embedding doesn't come from the class of Gaussians. Okay, so The test is specific to some test statistic T at some significance level alpha. So both of these are choices that you can make. We can compute an observed test statistic value from a mini batch. Just treat M as the size of the mini batch. So then T star, our computed test statistic, is computed by a p-value, which indicates how extreme T star is under the null. This is review for people, but just bear with me. The smaller the p-value, the more evidence against H naught. And remember, H naught here is that Py comes from the class of Gaussians. So if we have a low p-value, that means we're rejecting normality. Right, which is kind of what I say. And so alpha is just we're evaluating T star with respect to some quantile of T under H naught. So why do this? Why not just use Wasserstein autoencoders? This formulation has some benefits. So first, we can actually do this. So there are a lot of goodness fit tests that have closed form that are differentiable, which we need if we're going to use this in a neural network paradigm. We also have interpretability. So the p-value allows us to interpret why normality was rejected. We can adapt mu and sigma over training. So as we train, the mu and sigma can change. And we can actually track that and use that if we want to use that for some purposes later. And multivariate normality via projections is unaffected by rank deficiency. So what that means is we're actually going to use projections. Typically want to use univariate univariate normality goodness fit tests. And so we're going to project this larger dimension down to a smaller dimension. And during that projection process, we can actually test whether or not the covariance matrix is singular. So we don't necessarily need a full rank, which is pretty cool because you have really no sense of how big you should make the latent space, the dimension of the variation autoencoder. If you make it too big, we could project to a lower dimensional space, at least in theory. Yes.
Like a couple of questions. First of all, are these latent dimensions independent by each other, or are they orthogonal, or
The covariance can be full rank.
okay, right?
And it can, it could be spherical. You could have off-diagonal elements.
And going back on the test, no, you are doing this test m times. How many times are you doing?
Oh, sorry.
this test?
That's the test statistic computed on a batch of size m. And so
You are not doing every time, but
we are going to do it every time. And that's what makes this relatively slow. But we have to do this every time, and I'll tell you why in a sec.
Okay.
Yeah.
And you don't have any problem of unbalance or to like
No, because we can control the batch size.
okay.
Yeah, we can just sample a random batch of whatever size.
Thanks.
Sure. Yeah, good questions. Thank you. Okay, so our original objective in FG here are just neural networks with parameters theta and phi. Original objective is to minimize the expected reconstruction loss. That's what D is. X is the data, the translocation times. Well, here it was images, and then F is the encoder, G is the decoder. So that's just the reconstructed X. Subject to the push forward notation. Never mind the push forward notation here. Just subject to the embedding coming from the class of Gaussians. And then we have our typical Tikhonov reformulation using Lagrangians down here. So we're going to minimize the reconstruction, plus minus, depending on what test statistic we're using. So that really just depends on the test statistic. The test statistic computed on a batch of size m. Here's this is just setup of the autoencoder framework. We have parameters Cassie. Then we have this special set of parameters, capital theta, I'll describe in a second, and then phi for the decoder. Okay, so I'm rewriting the mini batch loss up here at the top. So that's just the same reproduced. So it turns out that optimization of this objective function suffers from some issues, some identifiability issues and some unbounded gradient issues. And one way you can think about this, at least in part, is you're optimizing the encoding to come from a class of Gaussians, but the neural network could just multiply everything by 10 or add 5, right? You can just scale the embedding of the Gaussians however you want, and it's still going to come from the class of Gaussians. So the way that we handle this problem is we restrict theta, which are those weights that are coming into Y, to come from the Stifel manifold, to be on the Stifel manifold, which is a compact Riemannian manifold of orthonormal matrices. So that's going to prevent this arbitrary scaling problem from actually affecting training. And so then we have our set of parameters. Those were just the set of parameters for the encoder and the decoder with our special Stifel manifold parameters. This forms a product manifold, which is Riemannian. And there are convergence proofs for Riemannian SGD, which we're going to use to train this thing, but they're not applicable to goodness of fit tests. So I would say probably the main theorem of this iClear paper is that we can still do Riemannian SGD and guarantee convergence. It's mostly the work of Aaron, but we're going to this Bonneville paper, which proved general convergence, and we showed that we don't need this uniform bound, I think, on the stochastic gradient. It's the way that we generalize to goodness of fit tests. Okay, the last methodological thing I want to show for this goodness of fit autoencoder paper is this principle of higher criticism. So this comes from Tukey. And you can think about this as like two-tiered hypothesis testing. So we have hypothesis testing at the mini batch level. Okay, that's going to tell us whether or not a mini batch is indistinguishable from normal. But then what can go wrong? So if we have a really high capacity F, and depending on what the regularization parameter lambda is, we could be pushing every single mini batch to normal, right? Would that be a good thing? It would not be a good thing, because if our null is that we have multivariate normality over all of the mini batches, we wouldn't expect every single mini batch to come from a multivariate normal, okay? If the null hypothesis is true and T is continuous, and we look at the distribution of p-values, those should be uniform over 0, 1, okay? So what we do is lambda was the coefficient in front of the hypothesis test, in front of the test statistic. We're going to observe a collection of p-values over training, and we want the collection of p-values to come from, if we look at the distribution of those p-values, uniform 0, 1. So we could have the case where the encoder is really, really strong and it's emphasizing reconstruction. So remember, low p-value is rejecting the null. The null is Gaussianity. So here we're rejecting the null too many times. Things do not look Gaussian enough. On the other end, if our lambda coefficient is too high, then we could be prioritizing Gaussianity too much. So things look suspiciously normal. What we want is this sort of Goldilocks zone where we do end up rejecting normality for some batches, but we have uniform p-values from 0 to 1. Okay, I'm just going to skip over the next result because I want to get back to the non-B problem. I just want to show one result from this paper. So what we do here is we track the parameters of the GOFE to compute a mean and covariance. And then we sample from that mean covariance, push it through the decoder, then push it back through the encoder and compute mutual information. That's the red line here. The blue is the KS uniformity test for a batch of p-values. Each blue point is a different KS uniformity test. The blue line is just the average. And the green is the condition number, the log condition number of the covariance matrix. So as that increases, the covariance matrix is being pushed to be more and more singular. And x-axis is just the log 10 of lambda. So what's really interesting here, right, is we don't know what lambda is. We want to try to learn what lambda is. As we increase lambda, okay, well, as lambda is really, really low, we're not encouraging the model to map to multivariate normality. We have high mutual information. It's doing a good job at reconstruction. Okay? Probably won't generalize well, but it's reconstructing well. But we don't have normality. We're rejecting normality too much. As we increase lambda, we see that the KS uniformity test starts to increase. So we actually do have uniform p-values. So we probably will generalize well. As we increase lambda too much, then we're pushing things to be too normal, and we no longer have uniformity of p-values. The p-values are too high. But there's like this sweet spot, right, where we have uniformity of p-values, and we still have high mutual information. So this performs, it's a principled way that's just based off test statistics for setting the regularization parameter. It's not based on cross-validation in the classical sense where you have something you're trying to optimize. Here, you have an interpretable way to set the regularization coefficient.
You
Yeah.
said that you could interpret when didn't meet the test, why you didn't meet the test.
Yeah.
So, okay, you did this with this auxiliary, you did this interpretation of this plot with this auxiliary data and many trials, but in general, if you look at this, you know, just at the two extremes, you have too low Kolmogorov-Smirnov test statistics. How much extra work do you need to do to interpret those seemingly similar situations?
Extra work for interpretation is not bad. You just actually look at the distribution of p-values, and it will almost surely conform to what you would expect. So if you're on the right side of this graph, then you're way too normal, you're overly normal. So the p-values are going to be bunched up around 1.
Fine, but then how does that lead you to an explanation?
Oh, so you're basically what that means is you're increasing the coefficient so much on your test statistic that you're forgetting about reconstruction of in the loss component, you're forgetting about the reconstruction loss. Okay? Or at least you're not prioritizing the reconstruction loss. So it's like you're taking, in the extreme, you would just be taking points and then mapping them randomly to multivariate normal and forgetting about, actually reconstructing them. You would typically do this if you're actually doing this, you would do this on a validation set, of course, not on the test set. So you do this on a validation set your lambda based off of some trade-off between uniformity of the KS values, which would indicate better generation, and mutual information, which would indicate better reconstruction. I said I was going to skip this, but that was a good question, and maybe I can show this really quickly. So like you can see that we have the KS uniformity test here for different test statistics, and we use Shapiro-Wilk, Shapiro-Francia, Kramer-Von-Misses, Kolmogorov, Smirnov, and Epps-Foley, I believe. And so different goodness of fit tests will work. Sorry, I shouldn't sing here. Different goodness of fit tests will focus on different aspects of the distribution. Some on moments, some focus on quantiles or tails. So that's why there's some variability here. But we see that Shapiro-Wilk and Shapiro-Francia do a relatively good job at reconstructing these images. This was done in Celeb data set of faces. And we also have uniformity. So as long as these KS uniformity test p-values are above 0.05, then we can claim uniformity. Whereas something like the beta VAE, which just places a weight on the KL term in a VAE, if you weigh that KL term way too high, then sure, you're really normal, but your reconstruction's terrible. Okay, let's go back to I'm sorry for the abrupt switch back to the original problem, but now we have this GoFA architecture. We want to use it for our problem. What does this get us? Well, we can train a model on just the BDNA. We can track the statistics of the mapping, right, of this multivariate normal, and then we can use that for hypothesis testing. So the way that we train this thing is we have a reconstruction loss. That's just the input translocation time and its reconstruction. I think we use L1, reconstruction error. On the purple, that's our test statistic in a spit. We just talked about that. And then there's this other component here. I should note that the X's are samples that are not labeled with a non-BDNA motif. I'm being a little bit careful there because it's not necessarily BDNA, but they're not labeled with a non-B motif. The Z's are labeled with a non-B motif. Okay, so there's this extra component that is just applied to the non-B motif samples. So think about, this is just a small cartoon to show you what this component of the loss is trying to do. So the A's are the BDNA, the B's are the non-B labeled points that have non-B structure, and the C's are the non-B labeled points that have BDNA structure, for which we think that most of them are probably BDNA. So you can see that we're trying to push the non-B structures off the manifold, and we're trying to reconstruct them poorly. So how do we actually do this? We have this extra component of the loss up in the top right. So think about what's happening if ZI is it's a non-B motif sample, but think about what's happening if it's actually non-B DNA structure. Then the distance between ZI and its reconstruction will be large, okay? Because we train this thing to reconstruct B DNA well. So if it's actually non-B structure, then that distance is going to be large, okay? So since we're minimizing, it's going to take away more from delta. Delta is a hyperparameter. Okay, and so that's not going to affect the loss too much. What if it's B DNA structure? If it's B DNA structure, then that distance between ZI and its reconstruction is going to be low, and we're going to incur a higher penalty. So this is, the second part is to encourage non-B structures to actually map off the manifold and reconstruct poorly. And we hope that the other part of the loss is going to be the one that's controlling B DNA structure, mapping well and reconstructing well. Okay, I'm running low on time, so we have some methodological notes that say that proves that we can treat this as a classifier and some large sample limit, but let me just skip that because I know you probably want to see some results. So the way that we actually make calls is we make calls based off of two statistics. F here is Mahalanobis distance, just the distance between a point and the multivariate normal that we estimate. So how far does the point map off the multivariate normal that we estimate from BDNA? And the other part is reconstruction loss. So if the point maps off the manifold and it's reconstructed poorly, it's probably non-B structure. So we calculate the null, the bottom here, from just BDNA, some held out BDNA set. So that's how we calibrate the null, and then we test on some additional BDNA and non-B samples. Finally, this will give us a bunch of p-values, and then we control FDR with BH. Okay, so does this actually work? I'm talking about a lot of method stuff, but does this work? Maybe there's nothing to be found. Just kidding, I wouldn't be here. So we evaluated some ONT data. This is NA12878, everybody's favorite reference genome. We extract translocation times 100 bases around non-B motifs. We have both the positive and the negative strand. And we use a validation set to learn a regularization parameter and to build the null distribution. So we have a nice null that we are pretty sure is coming from just BDNA. On the left is just the length of the non-B motifs. Y is the density. And so we can see most non-B motifs are small. I should say that this is in the old reference genome. So if you computed this again for the T to T data, it's probably shifted because we have way more repeats in that data. And then you see like the proportion of non-B DNA windows. Okay, so big takeaway slide. So these are quantile plots, very similar to the genome research paper. So you see the different quantiles and the different colors, control, which is BDNA, and non-BDNA in the solid line. And x-axis, position and window, the different facets are the different non-B motifs. So we see that for some non-B motifs, there really isn't too much distinguishing from BDNA, even at the upper quantiles. This could be, just because they're more rare. Maybe our quantile is not high enough. And maybe, quantile is kind of a mix of the different translocation times. So maybe there is signal, it's just not represented in the quantile plots. But for some of them, there's a very strong signal. And G quadruplex, again, the one that also stood out in the previous paper, there's a very, very high increase of translocation times in the strand that has the G quadruplex and a decrease in translocation times in the strand that doesn't. And this makes a lot of sense. If it's already single-stranded DNA, the helicase doesn't have to do much, so it flies through the pore. If there's structure, the helicase has to unwind the structure, and so it takes longer. Okay? And it's also kind of cool that, remember, the non-B is centered in the window. The signal is upstream of where the motif is. So there's like a lag between the signal and actually where the non-B motif is. Because bases are in the pore and the motifs here. So you have bases in the pore. Anyway. We compute a similar interval-wise testing that they did in the genome research paper. And this again is just showing you that we have increased, the signal is actually stronger than the genome research paper. We have significantly increased translocation times for G quadruplexes and even something like ZDNA. And even even for some of these non-B signals or non-B motifs that didn't have apparent signal, there's still arguably some signal there. Okay, then we the reviewers wanted to see some simulations, and so we generated some simulations with some functional form that resembled the non-B from the quantile plots. And then we tested both some classifiers and some novelty detectors. I mean, this is a new problem, and so there's no methods to do this. So we picked some off-the-shelf methods, showed that GoFA DND works pretty well. And then finally, like I said, there's one more slide after this, or one more group of slides. We can look in the experimental data at a specific FDR alpha level, 0.2, of how many calls does each method make. And GoFA DND actually makes the most calls most of the time. This isn't to say that all of these are actually true structures, but if you're prioritizing things for experimentation, you do want your method to make some calls. So besides G quadruplexes, it calls the most. Okay, and just I think I have 2 more minutes or so. This is, I think, one way you can interpret what's going on. And I really like these plots. This is the brainchild of Aaron. And so what you can see here is 4 different things. This is simulated data, so we know structure and motif. So the teal, the light blue, is DNA without a motif. So this is the embedding. We embed, and then we calculate those 2 statistics. Y-axis is reconstruction error, x-axis is Mahalanobis distance, okay? And so the light blue is DNA without the motif. That forms the null hypothesis, okay? And the more greenish color, that's non-rejected DNA with the motif. So it's non-B labeled, okay, but it's not rejected, meaning that it's probably B DNA, okay? It reconstructs well, and it's close, it's on the manifold. Then you have the square blue, which is rejected non-B DNA with the motif, true positives, and then red. And what is red? That's rejected B DNA with the motif. Okay? So these are false positives. But what can you sort of notice here? So this is, some people make this distinction, some people don't like making this distinction, but you can make a distinction between 2 classes of anomalies, outliers and novelties. Outliers are different from some normal class in a random way. Okay? They're just outliers. Novelties are different from some representation of the normal class in a specific way. So they're sort of clustered in some way. They have a pattern. So we can differentiate the false positives. They're outliers across, they're outliers on the tails of this distribution pretty much anywhere. I mean, there's an overabundance and the high reconstruction error and high Mahalanobis distance, but they're kind of scattered out throughout the entire. But the blue points, the actual true positives, have this tail, right? They have high reconstruction error and high Mahalanobis distance, and there's structure to them. So I don't think we did this, we call this in the paper, but call these comet plots, because you have this null distribution, and then you have this structure, which is not proof that they're non-B structures, but it's good evidence. I'm going to skip this. We also did some other non-B structures besides G quadruplexes. So just to conclude, translocation times do indeed seem to be associated with non-B motifs. We formulated the problem as a novelty detection. I guess there's other formulations you could do, but we thought this was the most relevant. And the way that we computed these non-B structures, putative non-B structures, are with this large scale hypothesis testing framework. Some caveats, future work. I'm sure that there's other sources of translocation time variation. We know that people call methylation from IPDs, interpulse durations, but we also know that that's related to non-B structure. So all your methylation calls from PacBio are confounded by non-B structure and vice versa. So is that also true for translocation times? We're working with some biologists to actually validate some of these calls in experimental samples. And we're also early stages of working with decode genetics to see if this is actually influential for disease. Tons of other methodological stuff as well. So thanks to the funders, data providers. Thank you for your attention. This is our group, and highlighted the 2 co-first authors of the work. Thank you. Any questions if you have them? Sorry, did I cut you off, Roberta?
What? Sorry.
Did I cut you off? I'm sorry.
It's okay.
I guess
You are doing this by yourself, so that's amazing.
ask one more question?
Sure.
You said you used the standard DNA in the bottle test. So did you use the trio or just one sample?
Just one sample, the child.
I see.
I have to check, but I don't know if all 3 samples had high coverage nanopore. I'd have to check, but we just used the child. And cats? Well, yeah. So we know non-B DNA exists, and we know that it's conserved across species. So we don't yet understand the role of non-B DNA and how it influences feline phenotypes. There's definitely no cat samples that have nanopore sequencing, unfortunately. Is there? There could be. It's really hard to. So one of the things that I glossed over is all the work that Marjan did, just the technical, boring software engineering work. But we need raw nanopore sequencing. We can't work with BAMs. We can't even work with Fast5s if it doesn't include the information on the current. And so even if you have raw nanopore, it doesn't mean we can actually apply this. So it was a pain. The genome in the bottle was really a godsend because that was the only thing I could really find. There's a couple of other that we're trying to use now, but we're working with the T2T consortium folks to do similar things in the T2T genome. I'm sorry?
Well, I guess we just put data because there are different enzymes.
Yeah, so the whole chemistry of how nanopore works, it changes slightly depending on what you're doing. But we and our biologist friends tell us that, well, it's a slightly different chemistry from that data set to this data set. Right now, we're not doing anything specific to the different chemistries. We just care about the frequency of current observations. But yeah, that'd be interesting, but we haven't so far done anything specific.
At least you have to assume that for your data can say things.
Yes, absolutely. And I mean, again, another thing I didn't really talk about is this is noisy. This is really, really noisy. So it's possible that different reads could actually be forming non-B or B. Okay, but there's no way you can make these calls on single reads. It's way too noisy. So we only consider windows that have at least 5 reads, I think, overlap, and we take like a median. So